In our last session, we have discussed about the development of your ventricular system along with the foramens, okay? And we have discussed about the histogenesis. So, what about histogenesis? What are all the structures we see in the histogenesis? We have seen the formation of neurosil, roof plate, floor plate and also lateral walls. After that, we have seen the formation of 3 cell layers. What are those? Matrix cell layer, mantle cell layer and marginal cell layer. So, your matrix cell layer is made up of, okay, mantle cell. Mantle cell is made up of neuroblast and spongioblast cells. The last layer is marginal layer. It is made up of okay. In this session, we'll start with the development of brain. So, in the histogenesis, we have already seen the formation of 3 cell layers. So, this is your neurosil, and the neurosil is lined by the pseudostratified columnar epithelial cells, okay, like this. And the next layer is mantle cell layer. These two together gives us the matrix cell layer. Okay, and the next layer is called as the marginal cell layer. Remember the matrix cell layer is also called as ventral zone, and the mantle cell layer is also called as intermediate zone, and the last marginal cell layer is also called as marginal zone. Okay, your neurosil is going to give rise for central canal part, okay, and your mantle cell layer, which is made up of spongioblast and neuroblast cells, it will give rise to your or forms your grey matter. Okay, the last one is marginal cell layer, it will give rise to your white matter. Okay. So, now we will see the formation of your spinal cord. This is the central canal, okay. This is the central canal. So, this area will be your roof plate, okay, and this is the floor plate. On either side of the central canal, we can see the lateral wall, okay. This is the lateral wall. Your roof plate and your floor plate are thin in structure, and your lateral wall is thick in structure. Your lateral walls are divided into dorsal part and ventral part. This is the dorsal part, okay, and this is the ventral part. This division of your dorsal part and ventral part will be done by this particular structure. It is called as sulcus limitans. This is called as sulcus limitans, Limitans. Okay, so your sulcus limitans divides the lateral wall into dorsal part and ventral part. The dorsal part is called as alar lamina, alar lamina, and the ventral part is called as basal lamina, basal, basal lamina. Okay. Your alar lamina is sensory in function, and your basal lamina is motor in function. Okay, is it clear for everyone? For the development of spinal cord, first we should study the histogenesis part. In the histogenesis, we can see the formation of 3 cell layers. And the 3 cell layers will give rise to different structures of your spinal cord. Here in the second picture, you can see the central canal, then we have the roof plate, floor plate. On either side, we have the lateral walls, and the lateral walls is divided into 2 parts by the sulcus limitans, that is called as alar lamina and basal lamina. Your alar lamina is sensory in function and basal lamina is motor in function. Okay. If it is sensory in function, which type of channels can be seen here or columns can be seen here? Afferent columns or efferent columns? Afferent or efferent columns? A or E? A. Okay. We can see afferent columns here. So in motor part, we can see the efferent columns. Is it right? Sure? Okay, fine. So in the sensory area, we can see the afferent columns, and in the motor area, we can see the efferent columns. In your spinal cord, we can see the 4 columns, okay, and all the 4 columns are general columns. General columns. Only in your brain we can see the special columns along with the general columns, but in your spinal cord we can see only the general columns. Okay. So in the spinal cord totally we have 4 general columns. What are those? 2 are present in your alar lamina part and 2 are present in your basal lamina part. Can you name them? This is general somatic afferent column and this is general visceral afferent column. Okay, and in the basal lamina, the first one or the inner one will be the general visceral efferent column and the outer one will be the general somatic efferent columns. These are the 4 columns we can see in the spinal cords. Any doubts? Any doubts in this? Sure? Okay. Remember, the somatic columns can be seen throughout the spinal cord. Okay. And the visceral columns can be seen only in the thoracic, lumbar and sacral regions. Remember this point, your somatic columns can be seen throughout the spinal cord, whereas your visceral columns can be seen only in the thoracic, lumbar and sacral regions. Okay. This whole concept is called as the development of functional components of the spinal cord. This is called as functional components of spinal cord. Okay, this particular diagram. Any doubts in this? Any doubts? So, how many columns can be seen in spinal cord? We can see 4 general columns. Okay, what are those? Okay. Okay, fine. So these are the 4 columns we can be seen in the spinal cord, and this concept is called as the development of functional components of the spinal cord. Okay, short note question. Then the next concept is positional changes of the spinal cord. Positional changes of developing spinal cord. Okay. So by the end of 8th week, we can see the complete development of your spinal cord. So in the 8th week, your spinal cord like this and your vertebral column. These two structures are present at the same level. Okay. After certain development or after differential growth, by the end of 24th week, your vertebral column will increase or elongated in the size, whereas your spinal cord will remain the same. Okay. We can see changes only in the caudal end. Okay. The cranial end will be same all the time, only the changes takes place in the caudal end. Remember this point. So at the beginning of the 8th week or at the end of the 8th week, we can see your spinal cord and also your vertebral column at the same level. But after certain changes or after certain time of development, by the end of 24th week, your spinal cord is present at the level of S1 vertebrae. This is vertebral column. Okay. And then by the time of birth, your spinal cord is present at which level? By the time of birth, your spinal cord is present at the L3 vertebrae level. Okay. Then after the birth, again we'll once check for the adult position or final position. Okay. So the adult position is L1 vertebrae. So in the adults, we can see the spinal cord at the level of, or the caudal end of the spinal cord at the level of lower border of your L1 vertebrae. Okay, remember the point, lower border of L1 vertebrae. This is the final position. Any doubts? So what is the importance of knowing the positional changes in spinal cord? At least why the positional changes takes place? Why the positional changes takes place in spinal cord? What is the purpose? Increase in height. Okay. Any other answers? Increase in the height of individual. Any other answers? Okay, once go and check why the positional changes of your spinal cord takes place. What is the purpose? Okay. Any doubts in this? So this finishes your development of spinal cord. For the development of spinal cord, first you have to start with the histogenesis of neural tube, followed by the development of functional components, then the positional changes. Okay. Then for the clinical anatomy, you can write the neural tube defects. What are the neural tube defects we learned in the previous session? There are two neural tube defects we have learned related to the anterior neuropore and posterior neuropore. Anencephaly and spina bifida. Particularly, we have discussed about the rhombencephalon, right? Hydrocephalus is related to your ventricular system, related to the brain part. Okay. So here you can write the defects of your neural tube, that is anencephaly and rhombencephalon, or spina bifida also you can write. Okay. Any doubts in the development of spinal cord? Yes or no? Any doubts? Okay. Histogenesis of the neural tube. The neural tube increases in thickness due to the repeated mitosis of its epithelial lining. By the middle of the 5th week of embryonic development, the transverse section of recently closed neural tube reveals 3 distinct layers, that is matrix zone, mantle zone and marginal zone. Okay. This picture we have already studied in the last session. You can concentrate on the sulcus limitans, and also here this is the dorsal part, so we can see the dorsal roots, and this is the ventral part which is present below the sulcus limitans, that is the ventral root areas. Okay. Development of spinal cord. Matrix zone is thick and lines the enclosed cavity called neurosil. Its numerous cells undergoing mitosis produces neuroblast cells and spongioblast cells. The former develops into neurons and later develops into the neuroglial cells. The neuroblast migrate to the adjacent mantle zone, the future spinal grey matter, and their axons enter the external marginal zone, the future white matter. Once histogenesis is complete, the remaining matrix cell differentiate into ependymal cells, and also it lines your central canal part. Development of the functional components of the spinal cord. On cross section, the cavity of the neural tube appears like a vertical slit. Due to the formation of vertical slit, the dorsal and ventral walls of neural tube remain thin and are called as roof plate and floor plates respectively. The lateral walls of neural tube will get thickened. On each side, the lateral wall of the neural tube is demarcated into dorsal and ventral region, that is the alar lamina and basal lamina. In few textbooks, it is also mentioned as alar plate and basal plates. Okay. And these two regions are divided by the inner longitudinal sulcus called the sulcus limitans. The cells of dorsal region or alar lamina are functionally afferent or sensory in nature, while those in the basal lamina are efferent or motor in nature. These are the 4 columns we can see in the development of your spinal cord. Okay, any doubts? This is the picture for the formation of your 3 cell layers. And here you can see the formation of sulcus limitans, that is dividing your dorsal and ventral parts. Okay, and also the formation of your central canal part. And E picture is the adult structure of your spinal cord, which you have already seen in your histology class. You can compare the structures with that ones. Any doubts? Okay. This is the picture for your functional component, which I have also drawn. You can see your notes, whatever you have taken the diagram, and compare the structures with it. Take 2 minutes and compare all the structures. So these are the columns which are representing in yellow, green, blue and orange color. What are this black structure? A thread-like structure which is running in this area. Nerves. Say it louder. Oh, okay. Any other answers? Where they are running? This column. Can you see this black structure, a thread-like structure which is running in the downward direction? Can you see this opening part? So I'm asking where those nerves or whatever the structures you are telling, no, that where they are going or for which structure they are running into. So what is the function of columns? What is the function of columns which we have discussed? Say it louder. Roots and signals. Okay, they are giving signals. They are giving signals to which part or what? What is the function of columns? Everyone. She is telling it will give signals. Any other answers? Okay. Stop talking, others. Okay. It acts as the formation of limbic system. It helps in the facial expression. hearing and breathing. Is that right? Okay, how the columns are related to the breathing part? How the column structures are related to the breathing part? Okay, once everyone just go and check your general anatomy part in the nervous system, what are the functions of columns, and also your neuron cells and the neuroglial cells, okay? In next class, I'll discuss about it in details, okay? When I'm discussing about the development of your brain, I'll discuss about that once again. Or you can check in your neuroanatomy textbook also, in the spinal cord part, okay? Next, we'll start with the development of your hindbrain. So, tell me about the brain vesicles, what we have studied in the development of brain vesicles. In which week we can see the development of brain vesicles? In which week? In the 5th week we can see the development of brain vesicles. So what are all the structures we can see there? At the primary or at the initial stage we can see the development of primary brain vesicles. We have 3 primary brain vesicles. Those are prosencephalon, mesencephalon and rhombencephalon. So your hindbrain is basically rhombencephalon part. Okay, and these 3 primary brain vesicles will give rise to secondary brain vesicles. What are those? So this is the telencephalon, diencephalon, mesencephalon, metencephalon and myelencephalon, followed by spinal cord. We have already seen the development of spinal cord, so we will study from the below to the above part, okay? So your rhombencephalon have 2 secondary cephalons. The first one is metencephalon and another one is myelencephalon. So your metencephalon will give rise to which adult structures? Your metencephalon will give rise to which adult structure? Medulla oblongata. Your metencephalon will give rise to pons and cerebellum part, okay? Your myelencephalon will give rise to the medulla oblongata part, okay? So the development of medulla oblongata is very similar to the development of your spinal cord. First we have to take this section at the myelencephalon part and see the histogenesis. It is similar to the structure of your spinal cord like this, okay? So here we have the neurosil. In the spinal cord part, the neurosil developed into your central canal, whereas in the myelencephalon, it will develop into which cavity? Medullary cavity. No, the cavity present in your cephalons region are called as the ventricles, right? So this will develop into your 4th ventricle. Okay, the neurosil will develop into the 4th ventricle. Later we have the mantle cell layer, then followed by marginal cell layer. For the development of your medulla oblongata, we have to concentrate more on the mantle cell layer part. Okay, we all know here in this region we have the sulcus limitans part. Okay, so during the development stage, your ventral part will get increase in the size because it will show the development rapidly. Okay, whereas the dorsal part shows the slow development. So it will remain as it is in the same, the size will remain the same. Only in the ventral part we can see the changes in the size. So once the changes takes place, this is how the structure will look. Okay. So this is the dorsal part, that is your alar lamina part, and this whole structure is the ventral part, that is the basal lamina part. Okay, and this cavity what you are seeing here is going to develop as your fourth ventricle, the floor part of the fourth ventricle. Okay, any doubts? Any doubts in this? Okay. First we have the histogenesis part of the myelencephalon. It resembles the same as your spinal cord structure. We can see the formation of 3 layers. In the spinal cord, your neurosil is going to develop as the central canal, but in the medulla oblongata region, it is going to develop as the floor of the fourth ventricle part. Okay. So here we know sulcus limitans is there. Below the sulcus limitans, it is called as ventral part, and above the sulcus limitans, it is called as dorsal part. So during the development, your ventral part will develop rapidly and it will get increased or become bigger in the size part. Okay, whereas the dorsal part will remain the same. It will show slow growth compared to the ventral part. Okay, any doubts? Okay. So now in this part we all know we have roof plate and here we have floor plate, right? So during the development we can see certain changes in the roof plate. For that changes we have to remember about the flexures. So what are flexures and how many types of flexures we have studied? What are flexures and how many types of flexures we have studied? 4 types. What are those? First tell me what is flexures. It's the bending. Why the bending takes place? Flexures are nothing but the bending of your developing brain part. Okay, so in the initial stage we can see the development of brain during the 3rd week, starting from the 3rd week. During that stage your cranial area is not spacious enough to occupy the developing brain part, so it will undergo the bending. Okay, so how many types of flexures we have studied? 4 types. What are those? Cervical flexure, mesencephalic flexure, pontine flexure and telencephalic flexure. So telencephalic flexure can be seen in this area, and then we have mesencephalic flexure which takes place in the mesencephalon area, followed by the pontine flexure which can be seen in your Rhombencephalon area. Then we have the cervical flexure which can be seen between your spinal cord and the hindbrain. Okay, so now this is the Rhombencephalon. Okay, and here the bending is taking place. Pontine flexure. Okay, so this pontine flexure will divide your Rhombencephalon into 2 parts, that is your metencephalon and another one is myelencephalon. So once the pontine flexure takes place, your roof part of the medulla oblongata region will get thin and it will also get elongated in both the directions like this. This is the roof part right now. This is the roof part. This will become thin and it will also get elongated in both the directions. Okay, I'll draw one more diagram for that. So once the roof gets, stop talking. Okay, so now this is the roof part which get thin and which also get elongated on both the sides. Okay, so once the roof get thin and it got elongated, your alar lamina will be pushed towards the dorsolateral side like this. Your alar lamina is pushed towards the dorsolateral side. Okay, so now this is the alar lamina. And this is the basal lamina. Any doubts? Any doubts in this? No doubts? Sure. So once the alar lamina get pushed towards the lateral side, the few cells present in this area will start moving in the upward direction and also in the downward direction. Remember, this is only the mantle cell zone. Below the mantle cell zone, again we have one more cell zone, that is the marginal cell zone. Okay, so whatever the cells from your alar lamina will now occupy the marginal cell layer like this. Okay, so this structure is called as the inferior olivary nucleus. Olivary nucleus. And the cells that move in the upward direction are called as the pontine nucleus. Since it is moving towards your pons region, they are called as pontine nucleus. Okay. So once the roof structure get elongated, your alar lamina is pushed towards the dorsolateral side. So once the pushing takes place, the cells present in your alar lamina region will start moving in both the direction, that is in upward direction and downward direction. The cells which move towards the downward direction will form the inferior olivary nucleus, and the cells that move in the upward direction will form the pontine nucleus. So this expansion of cells from the pontine region to your medulla oblongata region, this is called as bulbo-pontine expansion. This process is called as bulbo-pontine expansion. Any doubts? Any doubts in this? Say yes or no. Any doubts? Okay. Any doubts? For this one. The process of expansion of cells from the pontine region to the medulla oblongata region in the process of development of brain is called as bulbo-pontine expansion. Okay. So now we will see the functional components of the medulla oblongata. Can you name the functional components here? This is basal lamina. That means we can see So in the medulla oblongata on the alar lamina part, we can see 3 sets of columns, okay, and on the basal lamina we can see 4 sets of columns. And remember, in the hindbrain part we have both general columns plus special columns. Okay, any doubts? Any doubts in this? 3 columns in basal and 4 columns in alar lamina. Can you name them now? In the alar lamina region, we have general, somatic, afferent column and special somatic afferent columns. Okay, this is the second one. Third one is general visceral afferent column and special visceral afferent columns. Similarly, in the basal lamina part, we have the somatic. First one is somatic afferent columns, and the second one is general visceral afferent columns, and the next one is special visceral afferent columns. So in the medulla oblongata region, we can see total 7 columns, 4 in the alar lamina region and 3 in the basal lamina regions. The 3 we see in the basal lamina region are named as somatic afferent column, general visceral afferent column and special visceral afferent columns. And the 4 we see in the alar lamina are named as special and general somatic afferent column and special and general visceral afferent columns. Any doubts in this? No doubts? Is it clear? Cranial nerves are related to your medulla oblongata region then? Which cranial nerves are related to your medulla oblongata region? In the next class, we will study in detail about each and every column, their particular function or contribution to the development. Okay, so right now remember your 5th, 8th, 9th, 10th, 11th and 12th cranial nerves are related to this particular region. Okay, 5th cranial nerve, 8th, 9th, 10th, 11th and 12th cranial nerves are related here. So tell me the name of 5th cranial nerve. 5th cranial nerve is 8th one, 9th, 10th. Vagus nerve, which one? 9th, 10th, okay, 10th is vagus nerve. Others? 11th and 12th? 11th is spinal accessory nerve. 12th? Glossopharyngeal. Hypoglossal. Okay, 12th one is hypoglossal. What about the 5th, 8th and 9th? 5th one is? When I ask you, you can answer, okay? 5th cranial, 5th, 8th and 9th cranial nerves name. 5th one is trigeminal. 8th one? What about the 9th one? Glossopharyngeal. Sure? Okay, 8th one? Vestibulocochlear one. So the 5th one is? Okay, 8th. Vestibulocochlear. 9th one? 10th, 11th, 12th. Okay, fine. So regarding the cranial nerves and also the columns, we will discuss in the next class. Okay. Any doubts in the development of spinal cord and development of medulla oblongata? Any doubts? Sure, no doubts? Fine, then everyone write assignment on functional components. What exactly is functional components and what is the importance of functional components? Positional changes of the spinal cord. At week 8, length of the spinal cord and vertebral columns remain the same. The spinal cord extends along the entire length of the vertebral canal and the spinal nerve exists intervertebral foramen at the level of their origin point. Due to differential growth of the cord and vertebral column, the intervertebral foramen do not remain at the level of the spinal nerves. In order to exit from the corresponding intervertebral foramen due to the recession of the spinal cord, the spinal nerves are forced to go down in the oblique direction. So, what do you mean by recession of spinal cord? Recession of spinal cord. What do you mean by recession? The word recession. Going down. Okay. What do you mean by the word recession? Moving down. Moving away. Okay. Any other answers? Gap. Okay. So, in the initial stage, when your vertebral column and spinal cord are at the same level, your spinal nerves are corresponded to your particular vertebral columns or the vertebrae, okay. But once the positional changes takes place, your spinal nerves positions will also changes. Okay, in your spinal cord, there are few important structures called conus medullaris and cauda equina. So once we check for the adult position, this is your spinal cord and this is the vertebral column. And we can see the spinal cord at the level of L1 vertebrae, right? Lower border of L1 vertebrae. So whatever the spinal nerves present below the L1 vertebrae. So what spinal nerves we can see here? Below the L1 vertebrae? We can see lumbar, then what nerves we can see below the L1 vertebrae? We can see lumbar nerves, then sacral and these are the nerves we can see below the L1 vertebrae. So these nerves will start moving in the downward direction and also towards the lateral side like this. Okay, so this point, this is referred as cauda equina. Okay, remember this cauda equina, and also one more important structure is there, that is called conus medullaris. You can study about this in the gross structure. Right now remember this one, cauda equina. Okay. So at the 24th week, the lower end of the spinal cord ends at the S1 vertebrae. At the time of the birth, the lower end of the spinal cord ends at the level of L3 vertebrae. In adults, due to the further recession of the cord, they are mentioning once again about the recession. The lower end of the spinal cord ends at the lower border of your L1 vertebrae. The nerve roots that descends below the lower end of the spinal cord, the conus medullaris, try to remember the name, later you can study about this in your gross part, okay. Surround thin thread-like prolongation of pia mater from tip of conus medullaris to form the cauda equina part, okay. So this is the diagram for your positional changes of the spinal cord. You can compare all the 4 structures with all 4 weeks. The first one for the 8th week, followed by 24th week, Then during the time of the birth, the last D structure is the adult position. Okay. Okay. Next we have the development of brain. Once after completing about the nerves and the columns, I'll discuss about the nodes. Okay. Any doubts? Okay. Then we'll end the session here. Thank you.